Causality 2. This lecture consists of 4 parts. In the first part, I introduce the idea of potential outcomes as a useful way of pinning down more exactly what it might mean to say that one thing causes another. In the second part, I review some terminology that is widely used in causal analysis, but which is sometimes used inconsistently and therefore is prone to create confusion. In the third part, I discuss different types of treatment effect and why we might want to distinguish between them. Finally, in part 4, I briefly introduce some basic ideas about directed acyclical graphs, or DAGs. This is really a trailer for lecture 4, where they will be put to much greater use. Throughout this lecture, I draw rather freely on the exposition and examples used by Stephen Morgan and Christopher Winship in their excellent book Counterfactuals and Causal Inference. Sometimes ideas about causality are expressed in terms of an observed outcome and a counterfactual outcome, because what we are aiming at is a comparison between what actually is observed to happen and what might have happened had things been different. Ex ante, before we make our experimental intervention, or before the subjects are exposed to the treatment, neither the outcome in the treatment condition nor in the control condition is observed. Therefore, it's more exact to say that what we are dealing with are potential outcomes. In the simplest case, each subject has 2 potential outcomes: the outcome that will be observed if they are assigned to the experimental condition, and an outcome that will be observed if they are assigned to the control condition. Of course, ex post, only one of these is actually observed. The language of potential outcomes gives us a completely general way to think about causal inference that is applicable not only to experimental investigations, but also to purely observational studies. Let's begin by setting out some notation. First, we define potential outcome random variables, capital Y superscript 1 and capital Y superscript 0. Capital Y superscript 1 is the potential outcome in the treatment condition, and capital Y superscript 0 is the potential outcome in the control condition. It's crucial to understand that all subjects or cases have both potential outcomes. The realized values, meaning the values for any given individual drawn from the distributions capital Y superscript 1 and capital Y superscript 0, are denoted lowercase yi superscript 1 and lowercase yi superscript 0 for individual I. We can now define the individual level causal effect of the treatment. This is delta I equals lowercase yi superscript 1 minus lowercase I superscript 0. In other words, delta I is the difference for individual I in what would be observed if they were exposed both to the treatment and to the control condition. So, we've defined what it means to talk about an individual level treatment effect. But we're not done with the definitions. We need now to define some notation for exposure to the treatment conditions. Let's define a causal exposure variable, capital D, as follows. D equals 1 for those I actually exposed to the treatment state, and D equals 0 for those I actually exposed to the control state. So the observable outcome variable is defined as capital Y equals capital Y superscript 1 if D equals 1, and capital Y equals capital Y superscript 0 if D equals 0. This then brings us to the crux of the matter, the fundamental problem of causal inference. This problem is illustrated in this 2 times 2 table. In the rows of the table, we have the 2 possible states of the treatment variable. Cases are assigned either to the treatment or to the control state. In the columns, we have the 2 potential outcomes for each of the cases. The outcome that would be observed in the treatment state if the case were assigned to it, and the outcome that would be observed in the control state if the case were assigned to it. In any actual experiment, only 2 of the cells are observed. These are the potential outcomes in the treatment state for those that are actually assigned the treatment, and the potential outcome in the control state for those that are actually assigned to the control state. The other 2 cells are not observed. They are ex post as well as ex ante counterfactual. In an ideal world, we would like to compare outcomes within columns, but that is exactly what we cannot do. In a nutshell, the problem of causality is a problem of missing data. All we can do in practice is compare outcomes on the main diagonal, and that means we have to be interested in the details about how cases are assigned to the treatment and the control group. The standard move is to abandon the attempt to directly study the individual level treatment effect, which is unobservable, and focus instead on the average treatment effect, which, given the adequacy of our assumptions about the allocation to treatment mechanism, is observable. The first thing to do is define an average treatment effect, or ATE for short. We do that by using the expectation operator capital E, which we can just interpret as instructing us to take the mean over all the probability weighted values of a distribution. So, if delta I is the difference between the 2 potential outcomes for each case, then E, expectation delta I, within brackets, denotes taking the mean of these. But we know that we can't do that, because for each case, one or the other of these is unobserved. Nevertheless, we can exploit a trick in linear algebra. The expectation of the difference between 2 random variables is identical to the difference between the expectation of the first and the expectation of the second. And this difference we can examine, because we have some cases that are exposed to the treatment, and we have some that are exposed to the control. That will give us an estimate of the average treatment effect. However, and this is important, whether this difference gives us a reasonable estimate of the average treatment effect or not depends on how cases have been allocated to the treatment and control conditions. This is something that we will have to explore in much greater detail. It will turn out that if, as in an experiment, we know that cases have been randomized to treatment and control conditions, then we're in good shape. If, on the other hand, we are carrying out an observational study and have to take whatever nature throws at us, we will want to know much more about how the allocation came about before we can conclude that what we have actually estimated is the average treatment effect. Let's go back to treatment assignment in an RCT. Subjects are allocated to the treatment condition, D equals 1, or the control condition, D equals 0, by a lottery. Imagine we use a random number generator to create for each subject I a value in the interval 1 to 100 of a variable which we'll call capital R. Subjects are then allocated to treatment conditions according to the simple rule: if R is less than equal to 50, then D is equal to 0, and if R is greater than 50, then D is equal to 1. Here is how this works out for 4 hypothetical cases. Case 1 draws 23 and is assigned to the control group. Case 2 draws 31 and is also assigned to the control group. Case 3 draws 56 and is assigned to the treatment group, and so on. We know that R cannot have a causal effect on an outcome, capital Y, because R is just a random number. Whatever relationship R may appear to have with Y must be because it controls allocation to D. In a randomized controlled trial, we know how treatment allocation is assigned. It's assigned in a way that can have no impact on the outcome. In an observational study, we don't know that, so we have to learn as much as we can about the allocation process. If assignment to D is by randomization, or something that approximates to randomization, then we can say that the process is ignorable. The practical implication is that we need know nothing more about it or do anything in the way of statistical adjustment because of it. Another circumstance in which the treatment assignment mechanism is ignorable is when assignment to D depends only on a set of observed variables. Let's call that set capital S. In this case, we say that the potential outcomes are independent of D, conditional on S. This implies that correct inference about the effect of D on Y requires that in some way or other the variables in S, including their interactions, must be controlled for. The judgments we make about ignorability delimit the sort of research strategy or design that it makes sense to use. Ignorability through randomization under the investigator's control leads rather naturally to randomized controlled trials. Ignorability through randomization via the employment of a suitable instrument leads to what are sometimes called natural experiments, and by making the appropriate assumptions to instrumental variable estimation with observational data. This is not something that you need worry about right at the moment. I'll say no more about the latter right now, and we'll return to it later in the lecture sequence. Ignorability through knowing the variables that are responsible for selecting cases into treatment conditions leads, in the absence of randomization, to designs in which observational data are analyzed by conditioning on control variables, for example through the use of regression techniques, or to matching in order to create balance, or to forms of probabilistic matching, for example through the use of propensity scores. The take-home from this is that to make sense of a causal claim, you need to know, or be prepared to make reasonable assumptions about, how cases are assigned to treatments. There is also something else we should be aware of. In the social sciences, we're dealing with the behaviour of human beings. Humans do things for reasons. Some of these reasons have to do with their expectations about the future. 18-year-olds who doubt that they will do well at university are less likely to enrol. The unemployed, who are confident about their chances of getting a job, will not find a training program as attractive as those who feel their lack of skills set them at a disadvantage. If subjects are self-selecting into the treatment and control conditions, you can be fairly sure that one of the things that they take into account is an estimation of the future benefit of doing so. Mostly this estimation is private information, which is unlikely to be available to the investigator. The fact that humans make choices for reasons is one of the major challenges to making causal inferences when all we have to go on are observations on what nature gives us. So far, we might have proceeded, when discussing RCTs, without mentioning a key assumption we make when using one to make a causal inference. This is the so-called stable unit treatment value assumption, usually abbreviated to SUTVA. SUTVA is the assumption that the value of the outcome for an individual I exposed to treatment D does not depend on the way in which the individuals are assigned to treatments. Consider this hypothetical example taken from the Morgan and Winship book. On the left-hand portion of the table, in the column headed Treatment Assignment Patterns, we have in the first row the three different ways in which three cases can be assigned to the treatment and control groups, so that one case gets the treatment and two cases get the control condition. In the right-hand portion of the table, under the column labelled Potential Outcomes, are listed the potential outcomes for three cases. Case 1 is assigned to the treatment condition in the first allocation, and a score of 3 is observed. If they had been assigned to the control condition, as in the second allocation, they would have been observed to get a score of 1. The point to get here is that when only one case is allocated to the treatment condition, the difference in potential outcomes is 2 units. Now consider the second row, in which 2 cases are allocated to the treatment condition. Here, the difference in potential outcomes is only 1 unit. If this kind of situation existed, it would be a clear violation of the SUTVA condition. How might SUTVA be violated? Consider how we might move from the situation depicted in the first row of the table to what is being depicted in the second. To make things concrete, let's imagine that what is being evaluated is a program to promote employability skills among the unemployed. It consists of CV coaching, interview practice, advice about self-presentation, and so forth. The outcome might be the number of job offers received. The treatment is the sort of thing that it's difficult to prevent the control group from receiving. As the number in the treatment group increases, it's possible that there's going to be a spillover as friends pass on tips and information to each other, which implies a contamination of the control group, who in effect get some of the treatment. The consequence is that the treatment appears less effective than it otherwise would. This is in essence a threat to the internal validity of the experiment. Now consider a violation of the SUTVA assumption which is not so much consequential for internal validity as for external validity. Say hypothetically we run an RCT on the benefits of a job training program in a particular labour market. Only a very small portion of the working age population are involved, and the results suggest that those who enrolled in the program get significantly higher wages than those that didn't. Now imagine that we think it is such a great thing that we roll the program out to a significantly larger proportion of the population. Would we expect to find the same significant wage gains if we flood the market with skilled labour? It doesn't seem very likely. Humans live in social and economic systems with interconnected parts. Change one thing and the implicit ceteris paribus clause does not necessarily hold anymore. We literally can't always hold all things the same. This implies that the results from an RCT are often not scalable upwards. If, in a medical trial, we find that a new drug is better than the existing treatment, then rolling it out to everyone is unlikely to reduce its efficacy. Though even here we can think of some counterexamples, for example, the use of antibiotics. In the social world, things are not quite so simple.